Discussed about the arteries. Mainly we've discussed about the muscular arteries and elastic arteries. Okay. So only one portion is left, that is about the veins. Okay, slide of veins are also there, so you have to identify the slide of veins. Okay, now as I said, if you take any blood vessel, these basic three layers will be there. What are they? Tunica intima, tunica media and tunica adventitia. Okay, these three layers will be there. So same thing is present in case of vein also. Okay, but there are some differences that you have to know what are the differences present. Next, in case of vein, if you see the vein, there are different types of vein. Okay, that also we will see, and finally the structure of the vein, and then differences between arteries and vein. Okay. So everyone, you know what is the function of the vein. Okay, and based on the diameter, you can see the venules are there, then small vein, medium size vein and large vein. This is the classification depending on the diameter. Now if you see the smallest one, that is venules and small veins, these are the smallest vein, are also called as venules. Now they collect blood from the capillaries. So today morning first I have shown you how the blood is flowing, coming out from the heart, how it is going to the heart, if you remember. Okay, with the help of a flow chart. Okay, so in that I have shown you the capillaries. Okay, so the venous blood which is going from the capillaries, okay, in that first they will go to the venules. Now, the walls of the venules have considerable permeability, and it allows exchanges between the blood and surrounding tissue. Okay, that means whenever the blood is flowing through those venules, their walls are permeable. Okay, and so that the exchange of the material can take place. Now the venules are the sites where lymphocytes and other cells may pass from the bloodstream, and then it can reach into the interstitial of the cell, or the spaces between the cells or spaces between the tissue. Okay, diameter is less than 1 millimeter. Now the venules which are there, two types of venules are there: post capillary venules and muscular venules. Okay, two types of venules are there: post capillary and muscular venules. If you see the post capillary venules, these are microscopic. Okay, 10 to 50 micrometer in diameter. They receive blood from the capillaries, and they are lined by endothelium cells and pericytes. Pericytes are very important cells because they control the flow of blood through the capillaries. Okay. Next, muscular venules are there. You can see they are quite larger, 50 to 100 micrometer in diameter, and they are lined by endothelium. 1 to 2 layers of smooth muscle cells are present. Okay. Here you can see the venule. Okay, this is the arteriole and this is the venule. Okay. Next, large veins are there, medium veins are there. Okay. Now in case of large vein, again all these three layers are there, and they are well differentiated. If you take any large vein and if you see under microscope, you can see all the three layers, and it can be differentiated easily. Okay. So tunica intima, tunica media and tunica adventitia are well differentiated. Okay. Now in that, the tunica adventitia, that is the outermost layer, always thicker than tunica media. This is the most important feature in case of vein. Okay, if you take mainly the larger vein, in that case you will notice that the tunica adventitia which is there, okay, that will be larger as compared to the tunica media in case of vein. This is the most important striking feature of the vein. Okay, next, the walls of the large vein have elastic tissue, resist high pressure. Okay, for example, inferior vena cava, superior vena cava, etc. These are the examples of large vein. Now medium size veins, these veins also have three layers, but it's very difficult to distinguish. Okay, because the size is very small. For example, you can see splenic vein, testicular vein, etc. Okay, so in case of medium size vein, these layers are there, but mainly the layers, whatever they are present, it is very difficult to distinguish between the layers because of the smaller size. Okay, now here you can see the comparison. In case of medium size vein, poor identifiable three layers are there, and the diameter is 1 to 10 mm in diameter. Now tunica intima consists endothelium, basal lamina, thin subendothelial connective tissue and discontinuous internal elastic lamina. Okay, so all these three layers are there, but there are some changes. Now, what is the change in tunica intima, the innermost layer? The internal elastic lamina is there, but it is discontinuous in nature. Next, in case of tunica media, thin and few layers of smooth muscle cells are there. Okay, very thin, the tunica media. And adventitia is very thick, containing collagen fibers and elastic fibers. Okay, and most of the cases, the medium-sized veins, they are accompanying the medium-sized arteries. Okay, next, another important point which you have to note, that in case of medium-sized vein, okay, the valves are present. The medium-sized veins which are there inside that, valves are present, which is maintaining the unidirectional flow of blood. Have you heard about DVT, deep vein thrombosis? Forget about that deep vein thrombosis. Have you studied about the venous drainage of lower limb? So at that time, I hope you have three sets of veins are there: superficial, deep, and perforating. Then I hope you have studied about the valves also. So I am talking about that valve. Okay? So medium-sized vein shows presence of valves in their lumen to prevent the retrograde flow of the blood. That means unidirectional flow of the blood is maintained with the help of the valves which are present inside the medium-sized vein. Okay, why? Because valves are more in veins of the lower limb. For that, because the blood has to go against the gravity. In case of lower limb, the blood has to ascend against the gravity. So for that, these veins are helping. How they are helping? They supports the column of blood. Okay, and this thing already you have studied, and I hope you have studied about the peripheral heart. You have not studied which is peripheral heart? Yes. Okay, now here you can see the medium-sized vein. This is the cut section of the medium-sized vein, where you can see the tunica media. Okay. First of all, see the endothelium. This is the endothelium. Then you can see the tunica media and see the tunica adventitia. Okay, so the tunica adventitia is very thick as compared to the tunica media. Okay, next here you can see the valves of the vein. Okay, so the valves which are present inside the lumen of the vein, it controls the unidirectional flow of blood. That means it will prevent the retrograde flow. Blood will flow in one direction only, that is towards the heart. Okay, and that is ensured by the veins, valves which are present inside the valve. Okay, now the valves which are there, most of the vein contains valve that allow the flow of blood towards the heart. Okay, but prevent regurgitation in the opposite direction. Means the blood should not flow, because it is going against the gravity. So due to the pull of the gravity, it can go in opposite direction, retrograde direction. So that is prevented by the valves which are present inside the vein. Typically, each valve is made up of two semilunar cusps. I hope you have studied the heart. So in heart, you have not studied about the valves. How many types of valves are there? Two types of valves are there: atrioventricular valves and semilunar valves. Not bicuspid, tricuspid. These are the examples. ठीक है? Heart में दो type का valve होता है: atrioventricular valve and semilunar valve. Semilunar valves are present in the opening of the pulmonary trunk and aorta. And atrioventricular valve, that is tricuspid and bicuspid or mitral valve. Okay, so in that you have seen the cusp. So similar structure are present over here, and these are semilunar shaped. That's why these are called as semilunar cusp. Okay, each cusp is a fold of endothelium. Okay, and along with that some connective tissue are there, and elastic fibers are also present. Now valves are absent in very small veins. Okay, the veins within the cranial cavity or within the vertebral canal, and in the vena cava, these are also absent. Okay, mainly you can see valves are present in the veins of the lower limb. Next, the large vein. If you take the cross section, then as I said, these three layers will be present. Okay, the tunica intima is there, tunica media and tunica adventitia, and you can compare. Okay. So first, I hope you can see the endothelium. So this is the tunica intima, where endothelium is there. After that, basal lamina is present, and along with that, subendothelial connective tissue is there, but it is very thin. Okay. Next, you can see this particular layer is the tunica media. It is relatively thinner, and few smooth muscle fibers, fibroblast and collagen fibers are present. Now, most of the part is made up of, you can see, tunica adventitia. Okay. where what is present? Longitudinal smooth muscles. As you can see, longitudinal smooth muscles are there. Along with that, collagen fibers and elastic fibers are also present. Okay, so this is constituting the large vein. Okay, now here this is the difference between the artery and the vein. So artery we have discussed today morning, veins we have discussed just now. Now here is a comparison. Now how you will compare, or how you will make the difference in between this particular feature. So first, the wall, you can see the thicker wall of the artery is thicker. Whenever you are going to the dissection, I will request everyone to feel the artery and the vein. Okay, whenever you are dissecting any part, you will be getting the blood supply. You will see the blood supply. For example, you are in abdomen now. Have you seen the abdominal aorta? Next, have you seen the branches? Celiac trunk, superior mesenteric, inferior mesenteric. Okay, then I will request everyone to feel it. Okay. After that, what you will do? Have you seen the portal vein? Then feel the portal vein. एक हाथ से portal vein को feel करो, एक हाथ से artery को feel करो. Then you will feel the difference. That is important. Okay. Then you will understand this too. Next, you can see the lumen, oval, circular. Okay, it is collapsed, irregular. Internal elastic lamina well defined in medium and small arteries, and it is absent or not well defined in case of vein. Okay. Next, tunica media, you can see thicker than veins. That means the tunica media which is there in case of artery, we have seen that it is very thick, especially in case of elastic artery. But in case of veins, they are very thin. Okay. Next, tunica adventitia consists of connective tissue. Here, connective tissue is also there, but along with that, smooth muscle cells, elastic fibers are present, especially in case of large vein. Now, all the three layers of the vessels are well defined. Here, all the layers it is not defined. Only in the large vein and in medium size vein you can differentiate. If you go to the smaller veins, then all these three layers cannot be differentiated. Okay, so these are the differences between the arteries and vein. Okay, so that is all about the vein. This portion was left which I couldn't take, so that's why I have explained right now. Okay, so now directly we'll see the slides. First we'll see the slides of, which slide we'll see first? Lung. First we'll see the slides of lung. Now what you have to identify? In the slide of lung, what you have to identify? Wait, one by one. Bolo. What you will identify in the slide of lung? Yes. Yes. So first you have to identify in that area where interpulmonary bronchi are there. Okay. So try to identify that. Along with that, you have to identify the terminal bronchiole. Okay. And in some cases, you will be getting the respiratory bronchiole also, which is extending. The extending part will be the alveolar duct, and then alveolar sac, and then the alveoli. Okay, these are the parts which you have to identify. Okay, now for your reference, let me show you the diagram. See this one. See this diagram carefully. Okay, so under microscope, this kind of appearance will be there. Okay, so this is the intrapulmonary bronchus. First you will identify this, then we will identify the terminal bronchiole. ठीक है, देखो appearance कैसे आता है terminal bronchiole का. Same appearance will be there under microscope. So terminal bronchiole, once you identify, it's very difficult to identify the respiratory bronchiole, but still we'll try. It will lead to the alveolar duct, as you can see. It will lead to the alveolar duct, and then you can see the alveoli. Okay, up to that much we have to identify under microscope. Okay, to identify this, the intrapulmonary bronchus. Can you see the intrapulmonary bronchus? क्या था ना लंग से